First of all, I would like to thank the organizers for giving us this opportunity to present the view of the Academy on genetically modified plants. So first, a couple of words about the Forum for Genetic Research. We are a group of the Swiss Academy of Natural Sciences, and our mission is to foster dialogue between science and society, and also between science and the political bodies. We are a broad group of people, you can see that here, with a different background. Most of us have a scientific background, but we also have a theologian, we have people that are doing biomedical ethics, and as I said, we are interested in promoting the dialogue, and that's actually why I'm here today. So what are the challenges for the agriculture? The goals for the Swiss agriculture, and they would be pretty much the same goals in other countries, I guess, would be food safety, which means having sufficient production, competitivity, meaning cost reduction and efficiency, environmental protection, and quality. So these are pretty much broad goals which are everywhere important. And this goes, of course, partly through agricultural techniques, farming techniques, but also goes through what plants are used, and can one use plants having better properties. And so, generally speaking, there is a desire to have plants with improved properties, which can be many different things. It could be resistance to drought, it could be resistance to disease, it could be improved nutritional properties, improved durability, better taste, whatever. Any property that could be useful. And this obviously can be achieved by conventional techniques, but could also potentially be achieved by gene technology as through GMOs. And new properties, and it doesn't matter how they are achieved, whether they are achieved through gene technology or through conventional techniques, they always rely on changes in the genetic material. They always rely on something happening in the genome of the plants that will bring about the new properties. So how are these achieved? So here I show a picture of a few things which we all eat and which we know very well. However, they did not always look like that. So this is the ancestor of the plant I just showed. This is the ancestor of corn, the ancestor of carrot, the ancestor of banana, of eggplant, or of broccoli. So you see that this is the initial plant we had thousands of years ago. And the conventional breeding made it such that we now have the plants which we have and which we eat every day. So what we eat every day is not what was in the nature initially. We selected, through thousands of years of breeding, we selected plants having the traits that are desired, which make them better tasting, bigger, there is more food to eat on the plant. And so all cultivated plants have been dramatically modified genetically by natural means, just conventional breeding. And obviously wild plants would not feed us. I guess, if we would have to eat that, that would probably not be sufficient. So just an example for corn. So this is on the left side you see the initial ancestor a few thousand years ago, and on the right side you see the corn that people are eating today. And you see that there has really been a dramatic change in that plant, and this is natural. This is just based on selection. This is based on mutations taking place naturally, and the farmers observing that perhaps a plant looks a little bit bigger and selecting that one for the next generation. And over a long period of time, you can go from the left to the right, and you can have the plants which we are eating today. So you could say that genetically modified which is the normal way, normal breeding way, could be gene technology modified. And the goal of plant breeding has always been to select new and beneficial traits by natural means. So higher yield, resistance against drought, or whatever. So the first step of plant breeding, we need to identify and to generate genetic variation. In the old way, in the traditional way, this happens naturally. But this is very rare. Mutations take place rarely. So people have actually resorted to ways to increase the diversity, to increase the generation of these events. And this has been done either with chemicals or with ionizing radiation, so radioactivity. And then you can select the traits by breeding, but you could also introduce these traits by selectively gene technology. So in a way, I would argue that modern gene technology is only a logical development of what has been done. And we can discuss about the technology, but it's just a logical development. It's not something that is completely different from what has been done before. Just as an illustration, this is what has been called mutation breeding. So here you see in the center where you see this red arrow, there is a source of radioactivity, which will generate many mutations in the plants that are around there, and this generates a diversity, and from there one can then, through conventional techniques, identify the mutations that could be beneficial. An example of a plant a product that has been made like that, the left one is the starting one, the right one is the mutated one. That looks better, that perhaps tastes better, but this is not gene technology. That's the whole point. You generate mutations, you select the trait, and you have at the end the crop, but this is not considered gene technology. And there are thousands of these plants. They are absolutely allowed. They are used in organic farming, in Europe, anywhere. So there is no discussion about this being gene technology, although the technique has introduced mutations through, in that case, radioactivity. It can also be done with chemicals. And because this is anyway the natural way, the normal way without any addition is just to wait for a mutation to happen, a mutation that could be useful. And so this is not gene technology. There are thousands of those, and we eat them. So what is the basis of gene technology? Life is DNA and genes. So whether human, animal, or plants, there is always cells, and in the cells there is chromosomes, and chromosomes contain DNA. And the DNA is the DNA. It's the same DNA in every species. The building blocks of the DNA are the same. So what is gene technology? It's the capacity to recombine the genetic material between different origins, from different origins, and reintroduce them into a living organism. And if the living organism is an animal cell or an animal, then you call this red gene technology, and it's mostly used in biomedicine. This is what I'm working on. I'm not working in the field of plants at all. Or if the organism you use, or the target organism, is a plant or a plant cell, now you talk about green gene technology. And this is not overlapping with medicine, but is overlapping with agriculture, with food, with culture, and creates some discussions. Why can we do gene technology? We can do it simply because DNA, no matter which organism we know on Earth, they all contain DNA, and the building blocks of the DNA are the same. And therefore, people can, mix and match and generate new possibilities. So here I show the milestones of gene technology at different levels. On the left side, you see the first domestication of wild plants, maybe 10,000 years ago or so, or a bit more even. Genetics, maybe 150 years ago. Mutagenesis, the realization that DNA carries genetic information. Then the DNA structures in the 50s, you see the the little, you see actually the DNA structure, the double helix. And then a few more things were discovered. Agrobacterium transformation, or the first commercial genetically modified plants, are indicated in mid-90s. And this, however, it's a powerful technology, and very early on, already in 1975, there was a moratorium. Okay, thank you. There was a moratorium at Aziloma, which is in Northern California, where the scientists by themselves decided that they had to think about that technology. They had to think it through because they realized this is very powerful. So by themselves, not from the government or not from any pressure. And that set the path for the development of the technology, and I don't think that there has been any real issues from that technology. You could call this gene technology 1.0. However, now there is a revolution in molecular biology which also will affect plant breeding. And this is what we have been already touching upon a little bit here, precision genome engineering or editing. And I'll just tell you a few words about that because this is, I think, important to understand. Also because of that, there has been a new discussion, spontaneous discussion, a year ago in Washington, D.C., in December 2015, an international summit on human gene editing, because people realize that the possibilities we have with these new technologies are amazing now, and therefore we need to think about it. So these new plant breeding techniques, there are many of them, and I don't want to discuss anything except the genome editing, CRISPR-Cas9, which has already been mentioned, because I think this is really important to quickly understand. In this genome editing, the idea is always the same. You have the DNA, which you see here, and you go or you target the DNA at a specific location. And we actually know how to do that for already quite a few years. However, with the CRISPR-Cas9, which is the lower one here, we can do it in an extremely easy way, and it's extremely precise. So that's the big difference. We could do that already quite a few years ago, but this was incredibly complex and incredibly hard to do, and therefore very few people would do it. So it was not viable. Now it's easy to do. What then happens? You get a cut at this position. And if you do nothing, you add nothing, this cut will be repaired by the cell. And this is exactly what you would call a mutation. This corresponds to a mutation the way it happens normally. You could get a repair and then it looks like before, or you get a repair and it's a little bit wrong and now you have a mutation. There is no foreign DNA, there is nothing added to what you started from. So that's one option. You can also give a template for the repair and now you decide yourself how it will look. But you still had no additional DNA. It's just the homologous DNA. So now you have, where the red dot is, you have now changed maybe one nucleotide to make it a little bit different. So that's one option. And the final option is now you put a template which contains something in addition, perhaps another gene or perhaps some element in addition. So these are the three options. But the point is the left and the middle options are like a mutation. They do not add any DNA. So you go through the whole process. At the end, there is no additional DNA into the plant or the tissue, the animal cell or whatever. There is no additional DNA. It's just like a mutation, except you've done it in the lab and you know what you've done. So many people, many academies, many organizations have looked at the safety of gene technology. All have found it to be safe. I would just like to mention a couple of words here about how science works. Science works based on consensus. Science works based on the work of a community. And you have always different views on things, but you need to have a consensus. You need to have reproducibility, and you cannot focus on the outliers. You have to focus on where the majority of the people working in the field, what they see and what they observe. And in every field of science, and this is not different from any other field, there are always people who think, well, it's not like that, or we don't agree. That's okay, that's totally okay. But it's just a small fraction. One should always see, and this is why the academies all came to that conclusion, because the body of the people working in that field do not see the dangers or the problems, and they agree on that. And you always have, that's why we can't say that the scientific community is divided. You know, if you think of medicine, vaccines, they are the most important medical measure people can have. Still, there are people who are against vaccines, including medical doctors who are against vaccines. Yet, for the population and for the medical community, vaccines are extremely important and are totally accepted. So I think, from the point of view of the academies, we are, the technology is safe. So I think there are many misunderstandings which are listed here. You know, gene technology is a type of agriculture. Actually not. It's not. Of course, it has been used a lot in, in the USA and in many countries, but it's not intrinsically a type of agriculture. Intrinsically, it's a technology to make a change in a plant that could be useful. That's all it is. The usage is a different story. It's anti-organic. No, it's not. But of course, this is used, and you know the examples. Gain-tech free sells. It's a good way of selling things. That's okay. I have no problem. But it's not intrinsically anti-organic. You could use for organic farming a plant that has been made by gene technology. There would be no problem. It would be combining, good practice like organic farming with a plant that is perhaps useful. And of course, coexistence could be done. We could have, gene-modified plants next to plants that are done, in a traditional way. This is not impossible to do. So there are GMOs. It's not only GMOs made by the industry. There are also efforts to make GMOs that could be away from the industry. One of such examples is the golden rice, which, however, has been hampered in its development. And many people have actually changed their mind on that. I mean, this is one of the founders of Greenpeace who changed his mind on that. Also, Mark Linas is another one who thinks he saw it wrong. I think, to finish, I would just like to conclude by saying that gene technology is widely used in the biomedical field. There it has proven to be safe, and there is a good acceptance of the technology. The technology is the same, whether it's red gene technology or green gene technology. What the difference is the usage one makes. And there, of course, one can discuss about it, but the technology in itself is not, is not dangerous. GMOs are not intrinsically bad or good. It all depends on what it is. I showed you the example of the new technologies now, where we can, by using that technology, make a single, nucleotide change, or 2 or 3, whatever, which would be exactly like a mutation that would occur naturally. This plant would have no other difference, if it's a plant, would have no other difference that these few nucleotides change in the DNA. This is something that could happen by a natural mutation. The only difference is that in that case, one would know that one has made that change, and one would know exactly it is there. And in the other case, you just don't know. You know that you have identified a mutation that is useful for the plant. So I think, and I'm not the only one, I think we don't want to say that this technology is the solution for everything. Absolutely not. The only thing we say is that this is a technology that can be useful. That's all we say. But from a scientific point of view, this technology is not intrinsically bad or good. It's a technology that is useful and could be useful. And what we would like is that there is a rational discussion on GMOs away from, you know, away from emotions and really stay to the science. With, in particular, with the new technologies, we can have GMOs that are really like naturally modified plants. So I'll just acknowledge some of my colleagues who gave me some of the material for this presentation. Thank you very much.